Students, good morning. Today we discuss about DNA replication. First one, what is DNA? DNA is expansion of deoxyribonucleic acid. It is made up of three components. First one, deoxyribosugar and phosphate and nitrogenous bases. It is a genetic material of every cells. It's defined a long molecules containing unique codes that give instructions for the synthesis of all body proteins. Next we move on the DNA replication. DNA replication is the process. It's a semi-conservative process by which DNA makes a copy of itself during cell divisions. And the first step in the DNA replication is to unzip the double helix structure of the DNA molecules. The two separated strands which act as a templates for making the new strands of DNA. In short, DNA replication is the copying of cell divisions or copying of cells. This image shows that the DNA replication parent DNA strand to synthesize the or copying the new DNA strand synthesize. First one, DNA primase enzyme. DNA primase enzymes, it acts as a RNA polymerase enzyme that is used to synthesize or generate RNA primers, which are short RNA molecules that act as a template for the initiation of DNA replication. So every nucleotides are bound to each other in strands and joined with a phosphodiester bond, forming a sugar phosphate backbone. They are form a bond between a third carbon atom and the deoxyribose sugar. It is made up of one sugar and designate the 3' to 5' atom of the another sugar and the next nucleotides on the 5' end. Any part of the sequence can be used to create or recognize its adjacent nucleotide sequence during replication. So that DNA fits within the nucleus by being closely packed into the tight coils known as chromatins. These chromatins contains to form the chromosomes during the cell division. Before the DNA replication, the chromatins loosen up, giving the replication machinery accesses to the DNA strands. This image are the chemical structure of DNA. So this image shows that the main components of the DNA structures, the sugar phosphate backbone, adenine, thymine, guanine, cytosine, and from DNA to proteins, DNA to RNA, and RNA to proteins. DNA to RNA, it's called transcription process. RNA to protein translation process. These are including the DNA structure of DNA replications. So already we know that DNA replication is a semi-conservative process where a parental strand template is used to synthesize a new complementary data strand using several protein elements which include enzymes and the RNA molecules. The DNA polymerase enzyme is a main one of the enzyme in DNA replication process. These enzymes for catalyzing and joining of deoxyribonucleotide 5' triphosphates DNTPS, forming a growing chain of DNA. Other proteins are also involved for initiation of the process and copying of DNA along with proofreading capabilities to ensure the replication process takes place accurately. Therefore, DNA replication is a process that produces identical helices of DNA from a single strand of the DNA molecules. So it's one of an essential mechanism in enhancing cell growth, repair and reproduction of an organisms. These are this image shows DNA replication process, parental DNA to copying the or synthesize the new strand of DNA. The mechanism of DNA, it consists of three major steps. First one, opening the double stranded helical structure of DNA and separation of the strand. Second one, priming of the template strand. Third one, assembly of the newly formed DNA segments. During the separation of DNA, these two strands are uncoiled and specific site as known as origins. Its origin mean start of the one of the place. With the involvement of several enzymes and proteins, they prepare the strand for duplication. At the end of the process, DNA polymerase enzyme starts to organize the assembly of the new DNA strands. These are the general steps of DNA replication for all cells, but they may vary specifically depending on their organisms and cell types. Enzymes play a major role in DNA replication because they catalyze several important stages of the entire process. DNA replication is one of the most essential mechanism of a cell's functions, and therefore intensive research has been done to understand its process. So this mechanism of DNA replication is well understood in E. coli, which is also similar to in eukaryotic cells. In E. coli, DNA replication is initiated at the R calculus short Rg, to which DNA protein binds with while hydrolyzing of ATP takes place. So next move on the DNA replication enzymes and proteins. DNA polymerase is one of the enzymes to used for the synthesis of DNA replication. DNA by adding nucleotide one by one to the growing DNA chain, the enzyme incorporates complementary amino acids to the template strand. The DNA polymerase are found in both prokaryotic and eukaryotic DNA replication process. They both contain several different DNA polymerases responsible for different functions in DNA replication and DNA repair mechanisms. Next one, DNA helicase enzyme. This enzyme involved in unwinding the double helical structure of DNA, allowing the DNA replication to commence. It uses energy that releasing the ATP hydrolysis to break the hydrogen bond between the DNA bases and separate the strands. This forms two replication forks on each separated strand opening up in opposite direction. At each replication fork of the parental strand must unwind, exposing new section of a single stranded templates. These helicase enzymes accurately unwinds the strands while maintaining the topography on the DNA molecules. Next, DNA ligase enzyme. This is the enzymes that join DNA fragments together by forming phosphodiester bonds between the nucleotides. Another one enzyme is a exonuclease. These are the group of enzymes that remove nucleotide bases from the end of a DNA chain. Another one enzyme is topoisomerase. This is the enzymes that solve the problems of the topological stress caused during unwinding. They cut or both strands of the DNA, allowing the strand to move around each other to release the tension before it rejoins the ends. Topoisomerase is also known as DNA gyrase in E. coli. Next one is telomerase. This is an enzyme found in eukaryotic cells that adds a specific sequence of DNA to the telomers of chromosomes after the divide, stabilizing the chromosomes over time. Next we move upon DNA replication steps. DNA replication steps are consist of three stages. First one initiation, second one elongation, third one termination. First we discuss about the initiation steps. This stage where DNA replication is initiated. Next DNA synthesis initiated within the template strand at a specific coding region site known as origins. The origin sites are targeted by the initiator proteins with recurrent additional proteins that help in the regulation process to form a replication complex around the DNA origin. These are the several origin sites on which DNA replication is initiated, and they are all known as replication forks. The formed replication complex contains the DNA helicase enzymes whose functions is to unwind the double helix, exposing the two strands which act as a templates for replication. The mechanism of DNA helicase enzyme is by hydrolyzing the ATP that is used to form the bonds between the nucleobases, thus breaking the bond that holds the two strands. Additionally, during initiation, DNA primase enzymes synthesize small RNA primers that kick-start the function of DNA polymerase. DNA polymerase enzyme functions growing the new DNA daughter strand. Next one, elongation step. This is the second step of DNA replication process. The DNA polymerase are involved across the new DNA daughter strand by attaching to the original unzipped DNA template strand and the initiating short RNA primer. The DNA polymerase is able to synthesize the new strand and matches the template and the addition for the nucleotides to the 3' end. One of the templates reads to 3' to 5' direction. The DNA polymerase synthesizes the new strand to the 5' to 3' direction, which is known as leading strand. The other template 5' to 3' dash is elongated is an anti-parallel direction by the addition of short RNA primers, which are filled with other joining fragments, forming in a new formed lagging strand. The short fragments are known as Okazaki fragments. The synthesis of the lagging strand is discontinuous since the newly formed daughter's DNA synthesis. Third step is termination. After the synthesis and extension of both the continuous and discontinuous strands, an enzyme known as hexonuclease removes all RNA primers from the original sites. These primers are replaced with the right nucleotide bases. While removing the primers, another type of hexonuclease proofread the new strand, checking, removing and replacing any errors formed during synthesis. DNA ligase enzyme join the Okazaki fragments to form a single anified strand. The strands to the parent strand consist of a repetition of DNA sequences known as telomers, which act as protective gaps at the ends of chromosomes, preventing the fusion of nearby chromosomes. The telomers are synthesized by a special type of DNA polymerase enzyme known as telomerase. These telomerase enzymes are catalyzes the telomer sequence at the end of the DNA. And completion of the parental and complementary strand coil into a double helical shape, producing two DNA molecules, each passing one strand from the parent molecules and one new strand. So this image included the leading strand and lagging strand of the DNA replication process, and the replication fragments parental strands to synthesize the new daughter strand. It's involving the leading strand, lagging strand, RNA primers and Okazaki fragment. These are involved in the synthesis of new strand. Leading strand is a 3' to 5' directions. These primers generate a short piece of RNA known as primer by the DNA primase enzymes. These primers binds to the 3' to 5' direction, thus initiating to the synthesis of new strand. This is called as leading strand. And another one is a lagging strand. This template strand 5' to it starts as a 5' to 3' direction. That is a discontinuous manner by the RNA polymerase. These sites are templates are used and otherwise synthesize the Okazaki fragments. It's also involved in the DNA polymerase enzymes, which adds the pieces of DNA to the strand between the primers. So DNA replication, each new cell is formed with own new genomes that enhance the heredity via reproduction cell divisions. DNA replication stress, unusual DNA structure, mismatched ribonucleotides, tensions arising from the concurrent mechanism of replication and transcription, inadequate availability of important replication factors, fragile sites of the replicating DNA strand, overexpression or constitutive activation of oncogenes in accessible chromatins. That's all about DNA replication process. Thank you, students.